Okay, in this session, I'm going to go through the principles of epigenetics. So looking at the main epigenetic phenomenon, which is DNA methylation, histone methylation, and histone acetylation. And then we're going to look at how these processes control normal cellular differentiation. We're going to look at things like X chromosome inactivation. And we're going to look at specific examples of epigenetics playing a role in disease processes. So just to quickly outline what I'm going to talk about in this session, we're going to look at the different types of epigenetic modifications. So I'm going to primarily be talking about methylation of DNA. So this is where the DNA itself becomes methylated at cytosine residues, and this results in gene silencing. And we're going to look at examples of that, such as the silencing of tumor suppressor genes in a cancer situation. We're going to look at epigenetic changes that occur post-fertilization. So after fertilization, DNA methylation patterns are lost from the gametes upon fertilization, and we'll look at why that happens. We'll then look at the next major type of epigenetic changes, which is your histone methylation. So we'll look at histone modifications, including methylations and acetylations. And we'll see how all of these are both heritable, as in when a cell divides, the methylation patterns can be or are predominantly passed on and heritable, whilst at the same time they are reversible and changeable and malleable depending on what signals the cell receives. Finally, we'll look at some epigenetic phenomenon, such as the role of the XIS gene in X chromosome inactivation. We will likely look at imprinted genes. So this, in particular, the Prader-Willi-Angelman syndrome region, which is all down to either maternal or paternal methylation patterns in gametes. And we will look at citrullination, which is required to change neutrophils into an important part of the immune system, which is neutrophil extracellular traps. And this is an epigenetic phenomenon because it involves changes to histones and therefore chromatin patterns. So before we go into too much detail on the epigenetic phenomena, we'll look at the basics of chromatin. So chromosomes are made up of chromatin, and that chromatin can be found in two major forms, and that is heterochromatin, which is what we call compact chromatin. And this heterochromatin tends to be found in patches throughout the chromosomes. It can be in the middle of the chromosomes, but particularly you'll find patches of this at the ends of chromosomes near the telomeres, so here and here, and particularly around the centromere. And this is because these regions are lacking in genes. The ends of chromosomes can become eroded. You don't really want your genes to be there. So these regions are compact. They are very sparse in terms of genes. But you also get regions of heterochromatin randomly in the middle of chromosomes as well. So this is pretty much silent chromatin. There aren't many genes within there. If there are genes in there, it's very difficult for those to become expressed. And then we have euchromatin. This is more gene dense. It has very relaxed chromatin. That means that the DNA is wrapped more loosely around histone proteins, and we'll come onto that phenomenon later on. And these regions tend to be more associated with unmethylated DNA and acetylated histones, and both of these things allow transcription to occur. So unmethylated DNA and acetylated histones tends to be the pattern where you've got particularly loose chromatin, and that allows transcription factors to access the DNA and switch on gene expression. But we're going to cover that in more detail in a moment. So before we go into too much detail about gene regulation, I thought this slide might be useful just to remind you about the basic anatomy of a gene. Now, an average sized gene, maybe 100,000 base pairs in length from start to finish. only a minority of that is coding for mRNA and protein. So the endpoint of almost all genes, most genes, is the production of protein, and that protein is coded by mRNAs. Those mRNAs are coded by the production of mRNA, which is the copying of the exon components of a gene. So if we look at this is the DNA sequence up here, and the DNA sequence associated with this gene doesn't just contain the gene coding exons, it also contains the introns, and it will contain the 5' and 3' untranslated regions. Now these regions form part of the messenger RNA, but they are not coded into protein. So for transcription, mRNA production, RNA polymerase will bind roughly here and make an mRNA copy of the DNA for that gene. That will include the 5' untranslated introns, exons, and the 3' untranslated region. And this is our mRNA. And this is a pre-mRNA, and then those introns will be spliced out, and that will give us our mature mRNA that will be exported from the nucleus and translated into protein by the ribosomes. So this is rather simplified, and if we're looking at the whole gene, we have enhancer elements here, and we have these upstream enhancer elements here. Now, RNA polymerase can only bind to the promoter region of a gene if transcription factors are present, and the transcription factors bind to specific DNA sequences called enhancer elements and proximal enhancer elements, and there are these downstream enhancer elements. Now, the transcription factors have to bind to these DNA sequences to allow RNA polymerase to bind, and those proteins that allow RNA polymerase to bind are transcription factors. Without transcription factors, you don't get transcription. Now, epigenetics, or some aspects of epigenetics, result in the change of DNA at the transcription binding site regions, and specifically what we're going to deal with first is DNA methylation, because if the DNA is methylated at a transcription factor binding site, let's say here, that may stop a transcription factor from binding, and therefore RNA polymerase cannot bind, and therefore transcription is silenced. Now first we're going to look at DNA methylation, and DNA methylation occurs at cytosine, specifically at cytosines and at no other bases. Now we have got, here's a cytosine, here's guanine, and DNA methyl transferase is the enzyme that can allow the addition of a methyl group, so a CH3 group, onto cytosine, and this is our 5-methyl cytosine. So this is methylated DNA, this is unmethylated DNA. And this here is S-adenosyl methionine, which is not really, just don't worry about this, it's not important at this stage. All you need to know is that DNA methyl transferase adds on the methyl groups at cytosines. Now what's really important is that these cytosines are always next to a guanine. So this is a C, this is a phosphodiester bond, this is a G. So this is called a CPG island. So it goes C, phosphodiester bond, G. So this is an unmethylated CPG island. This is a methylated CPG island. Now these CPG islands are absolutely crucial for controlling transcription. And so CPG islands are found within the promoter regions of genes, and they are particularly rich in CG dinucleotides. So this here just shows a region of DNA from the promoter region of a gene, and this shows a DNA sequence from a random bit of DNA. And all of the CG dinucleotides are highlighted in yellow, and here in red is the ATG start site, which is the translational start site. Now, translation normally starts at an ATG start site, which codes for methionine. So what you can see is these are very rich in CpG islands, and that means that the promoter regions can become methylated or they can be unmethylated if the cell decides that that gene wants to be switched on or switched off. And some of these are more crucial than others, but certain CpG dinucleotides sit right where a transcription factor is required to bind. Now this slide illustrates that in more detail. So here is the promoter region and the tartar box, and the region here where RNA polymerase will bind and will transcribe this region in blue. Now upstream from that, we have these enhancer regions where transcriptional activators or transcription factors will bind to specific regions of DNA. And the sorts of things that we could be looking at here are response elements, so estrogen response elements, EREs. It's a specific run of nucleotides that the transcription factor estrogen receptor binds to. Similarly, androgen receptor, the P53, they all recognize very specific runs of bases, and these are found in the promoter region of the gene. Now, these can be tens of thousands of base pairs away from the transcriptional start site. So what you tend to have is a whole region of DNA. You can have proteins that cause the DNA to fold back on itself. You've got these enhancer regions, you've got transcriptional activators, and they may be required for the recruitment of general transcription factors, which subsequently allows RNA polymerase to bind. Once everything is in place, RNA polymerase can bind and we get RNA synthesis. If one or more of these components here are missing, then the whole complex can fail to form, and the thing that can prevent those from these little orange activators or transcription factors, the thing that can stop them from binding is a single C turning into a 5-methyl cytosine at this point here or this point here, and that can stop the whole complex from forming, and that can silence this particular gene. And this is just shown in a slightly more complicated cartoon here, where we have got transcriptional start site. Here we've got RNA polymerase. We've got a whole complex of transcription factors and general transcription factors. We have histone modifying enzymes. We've got DNA methyltransferase. We've got a whole mass of proteins all controlling whether RNA polymerase is allowed to do its job or not. And this can be involving enhancer regions, which are thousands of base pairs away from the transcriptional start site, but are folded back upon the promoter region and linked together by transcription factors and enhancer proteins that will enhance gene expression. And as I said before, if this region here, or this region here, or this region here is methylated, these proteins will not bind and the whole complex will not form, and we will have gene silencing by DNA methylation. Now, after DNA replication at the replication fork, DNA, newly produced DNA, is initially unmethylated. So if you think back to your lectures on the replication fork and Okazaki fragments and all of that type of thing, you know that DNA is antiparallel. You separate that DNA at the replication fork, you get a leading and a lagging strand where you get synthesis of new strands of DNA. Now this is where CpG islands are really important, because if you've got a C and then a G and the C is methylated, then on the opposite strand you will have a G, then a C, and the C will be methylated. So both strands will be methylated, but only at the C's. But if there's a C next to a G, on the opposite strand there will be a C next to a G. When you separate those two strands, so helicase comes along, separates the two strands. Here is this bit here, that's methylated. We get a new strand synthesized, that is unmethylated. Here's this methylated bit of DNA, here's a new strand that is unmethylated. Now what happens next is that the DNA methylation patterns are maintained by DNA methyltransferase, it will come along, it will recognise the methyl there, and it will tag the cytosine opposite with a methyl group. So in that way, upon cell division, methylation patterns are heritable and maintained. Now there are situations where these methylation patterns are not maintained, and one of those examples is upon fertilization. So here's an egg, here's a sperm. Upon fertilization, there is a deliberate demethylation occurring, and if we look specifically at this graph here, and this is from fertilization to the point where you end up with first mitotic division, then we've got a two-cell embryo. At fertilization, let's say that all DNA is 100% methylated. Not that it's 100% methylated in total, but that is the background methylation level of sperm and egg DNA. Some genes are methylated, some are not. Now, immediately after fertilization, the male-derived DNA is deliberately demethylated. So that will go to very close but not quite 0% methylation. Basically everything is unmethylated apart from a few key regions which we will call imprinting regions, which we're going to cover right at the end of the lecture. The female derived DNA is passively demethylated. That is, when the cell undergoes cell division, the DNA methylation patterns are not maintained, so that as we go to the two-cell embryo stage, the female DNA is 50% methylated compared to what it was. When it undergoes cell division again, and you're up to the four-cell stage, it's 25%, and then down to 12%, and so on until it reaches a very low level of methylation at the 16-cell stage. Now what you can see is the transcription levels increase as DNA is unmethylated. And this slide just shows that process, showing fertilization occurring. We've got the paternal genome in, can just about make out that's blue, becoming deliberately, actively demethylated as the embryo goes from the fertilization through to the 8-cell stage, whereas the maternal DNA is passively demethylated. That is, it's just the methylation patterns are not copied across, so you go from 100% down to about 12.5% when you get to the 8-cell stage. And what you see here are, in this little dotted line, are markers of breakdown products of 5-methylcytosine, which we can detect in those first few days, showing that there is some changes to methylation status. I won't worry too much about those, but the point is that you can see those occurring. And then as the embryo moves through to the morula and the blastocyst stage, you then get 5-methylcytosine levels rising and occurring specifically in certain regions, and that is then controlling gene expression. So whereas here lots of genes are switched on, here we're selectively pruning and switching off genes to help shape the embryo. Now this slide, it's an experimental situation, but it shows just how profound the epigenetic changes are during differentiation. So this is an array assessing CpG methylation patterns in different cells and different tissues. And these cells here, these iPSCs, which are induced pluripotent stem cells, and also some embryonic stem cells. These are very undifferentiated cells. And these cells, you can see each green little bar on here is a different CpG island. If it's green, it is unmethylated. If it's red, it is methylated. So in these induced pluripotent stem cells, which are very primitive and able to form any cell type, most CpG islands are unmethylated and a few are methylated. When you start to get specific tissues, you see different patterns of methylation. So if we look here at placenta, we've got these cluster of genes here which are unmethylated, and almost everything else is methylated, whereas in different tissues you get different clusters. So this is just an experimental situation and clustering genes based on whether they are switched on or switched off in different tissues. Now that is been shown in the lab, and this is what's happening to help define gene expression patterns in different tissues. Now this patterning can go wrong in disease, and one of the main diseases where it consistently goes wrong is in cancer. So this diagram at the top is some normal colon, and blue is just the stain for the colonic epithelial cells. In brown is where you get the expression of DNA methyltransferase, and it tends to be expressed in these basal-like cells and possibly stem cells. So very few cells express DNA methyltransferase. Most of these cells are terminally differentiated into colon cells, and they are fixed as colon cells, but these are more plastic in their ability to change. This is a moderately differentiated colon cancer, and this is a very poorly differentiated, so this is a very aggressive type disease. And what you can see is there's a large amount of cells which express DNA methyltransferase. Now this is because the cancers are expressing DNA methyltransferase, and in doing so, aberrantly, they are then able to silence genes that do not benefit the cancer. So this is one tumor suppressor gene called the RASF1A gene. It's a tumor suppressor gene that's important in cancer and particularly colon cancer. And these circles here represent CPG islands around the promoter region. So this is exon 1, this is the promoter region. And what you can see is in normal control individuals, all of these CPG islands are unmethylated. When you look at the DNA from the tumour, the CPG islands are predominantly methylated, and therefore the RASF1A gene is silenced in cancer. Now the mechanism for this silencing is likely to be related to overexpression of DNA methyltransferase. Now this phenomenon is present in many tumour types, and there are many different tumour suppressor genes. Now, tumour suppressor gene is a gene that either controls and suppresses growth, or maybe alters cell survival, or regulates DNA replication and DNA repair. And the tumour will gain a benefit from silencing any of these. So these down here are all tumour suppressor genes, and these are different tissues. And when you look at the cancers from all of these different tissues, you see specific tumour suppressor genes which are reliably silenced in specific cancers. So these bars just represent the percent of DNA hypermethylation and hence gene silencing in those particular cancers. So we can see that in leukemias, that huge bar there is down to P14-ARF. We can see, for example, esophageal cancer, CDH1 is absolutely crucial, and that is almost predominantly almost always silenced by aberrant DNA methylation in esophageal cancer. So this is the end of part 1 of this lecture. Part 2 will be coming up, and that will be focusing on histone regulation. Now what you might want to look up is how DNA methyltransferase can be drugged and inhibited in a cancer situation, and think to yourself, what will be the purpose of doing that? And look up any drugs which are DNA methyltransferase inhibitors, work back through the slides that we've gone so far and think, how would that affect tumour growth, and what is the rationale for using those drugs?